Warm wishes from J. Albinovins, Assistant Professor of Botany, Holy Cross College, Nagarkoil. Today, we are going to discuss about DNA fingerprinting. The DNA fingerprinting is a very quick way to compare the DNA sequence of any two persons or any two individuals. This technique was first described and developed by Alec Jeffreys, who is the recipient of world's oldest science prize, Royal Society's Copley Medal in August 2014. Actually, the DNA of person is unique in much the same way as the fingerprints are. We all know that there are 23 pairs of human chromosomes with 1.5 million pairs of genes. We all are aware that genes are the segments of DNA and they differ in the sequence of their particular nucleotides. As we know, not all segments of DNA code for proteins. Only some DNA fragments have regulatory functions, and the other DNA or intervening sequences. These intervening sequences are otherwise known as introns, and still some other fragments are repeated DNA sequences. And for this DNA fingerprinting, the short repetitive nucleotide sequences that are specific for a particular person are very, very important. These nucleotide sequences are known as variable number tandem repeats, that is commonly known as VNTR. The DNA fingerprinting technique involves identifying differences in some specific regions in DNA sequence that is known as repetitive DNA. In repetitive DNA, the small stretch of DNA is repeated again and again. These repetitive DNA can be separated from the genomic DNA as different peaks during density gradient centrifugation technique. The bulk DNA that forms the major peak and the other small peaks are known as satellite DNA. So, depending on the base composition, that is adenine with thymine rich or guanine with cytosine rich, the length of the segment and number of repetitive units, the satellite DNA can be classified into many other categories like microsatellites, mini satellites, etc. These type of sequences can show high degree of polymorphism and this form the basis of DNA fingerprinting. Let's see the mechanism of DNA fingerprinting. We all know that every cell possesses DNA. Extremely small amounts of blood in semen, hair bulb, or any other cells in the body of the person show the same degree of polymorphism, and they become very useful as the identification tool in forensics area. Further, as the polymorphisms are inheritable from the parents to the offsprings, the DNA fingerprinting forms the basis of paternity testing in case of disputes. The technique of DNA fingerprinting is accepted worldwide. Let us see the process of DNA fingerprinting. In the process, first we have to extract the DNA from the sample cells. After extracting the DNA, we have to amplify with the help of PCR, that is polymerase chain reactions. So during the amplification, we can get many copies of DNA. After getting the copies of DNA, that is after amplification step, we have to go for restriction digestion. So this digestion is carried out with the help of the enzymes known as restriction endonucleases. After that, the DNA fragments should be separated with the help of agarose gel electrophoresis. After the process of electrophoresis, southern blotting is done. During this process, the separated DNA sequences are transferred from the agarose gel into the nylon membrane. After the transfer, we have to go for hybridization by using VNTR probe. VNTR is nothing but variable number tandem repeats. So during this process, the radioactive DNA probes are attached to the specific portion of the DNA fragment. So thereafter, any DNA not attached to the probe can be washed off. So after hybridization, we can detect the hybridized DNA fragments with the help of autoradiography. So during this time, what happens? The remaining DNA is further exposed to the X-ray film, and the radioactive probes present on the DNA are allowed to expose the film, which when developed relieves the unique pattern of both dark bands and light bands. So these bands will give us the characteristic pattern for the individual nature of DNA. Actually, it differs from individual to individual, except in the case of identical twins. The sensitivity of the technique can be increased by using PCR reactions. Consequently, the DNA from the single cell is actually enough to perform DNA fingerprinting analysis. And recently, many probes are used to carry out these DNA fingerprinting reactions. So coming into the applications, mainly this DNA fingerprinting is used to identify the individuals with crimes such as raping cases, for settling the paternity disputes, to determine the relationships for immigration purposes, and also to detect the inherited diseases, and even we can monitor the bone marrow transplantation techniques. Beside that, it is also useful for determining the population diversities and also the genetic diversities.